Catabolism goes on. Now we are talking about respiration. Yeah, respiration. We have oxidized like hell in the citric acid cycle, and we have produced tons of reduction equivalents, tons of NADHs, tons of FADH. Where to go with it? If we go through the citric acid cycle, there is no internal electron acceptor which could use these tons of reduction equivalent. We need an external electron acceptor, and that's respiration. And let's look how we can produce, using respiration, energy. We have talked about the citric acid cycle, and we have talked about the net reaction here. Now the point is, if I look at this equation here, this is the net reaction. If I look at the net reaction of glycolysis and the citric acid cycle, this is at the end what I get. So I start with glucose, and at the end I produce 6 carbon dioxide molecules if I couple the glycolysis with the citric acid cycle. Why? The product of glycolysis are 2 molecules of pyruvate. Remember, the aldolase splits fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Dihydroxyacetone phosphate, of course, can be moved to glyceraldehyde 3-phosphate. So this is the same. So at the end, you produce 2 molecules of glyceraldehyde 3-phosphate. And now you go further and you produce 2 molecules of pyruvate. If you introduce these 3 molecules of pyruvate into the citric acid cycle, you will fully oxidize that. So you will get from 3 molecules of pyruvate 3 carbon dioxide molecules. So at the end, glycolysis plus citric acid cycle is a full oxidation of glucose. So you produce some ATP, GTP per molecule. Remember, per cycle, in the citric acid cycle, we produce 1 GTP per glycolysis per molecule glyceraldehyde 3-phosphate. We produce 1 ATP. As it's twice, we produce 2 GTP and 2 ATP. So the ATP and GTP production is not overwhelming in this, but what we produce is we produce tons of reduction equivalents. So in other words, at the end, we want to produce energy. We're talking about catabolism. And to be honest, our energy yield here, our direct energy yield with respect to the energy equivalents, what we have produced, well, it's 4, at the end it's 4 ATP. GTP and ATP is the same. So it's 4 ATP. That's not too much. So we're happy about that part. But we don't really know what to do with this part. So it's the same. So we cannot just, we have to produce now tons of reduction equivalents. If we would do the cycle again, we would consume all our NAD. So we need to get rid of this somehow. And we have produced a lot. So you remember, fermentation pathways are pathways where we use internal electron acceptors, but this doesn't work here. But now let's see, what is the energy content of this NADH? Well, NADH is nothing else than hydrogen at the end. So the reduction potential of NADH and hydrogen is merely the same. So if we transfer now these electrons from NADH onto oxygen, we burn, it's kind of burning hydrogen. So we produce a lot of energy. Now let's see whether we can exploit that. So let's go back. So essentially what we have is the following: our oxidation, which is this part reaction. So you might remember, this looks more or less the same as what we have done with the formation of water. So it's NAD plus H plus 2 electrons. Yeah, this is the oxidation part. And now I have my reduction part. And this is, of course, 2E minus. Now I just put also the protons, and we can directly produce our water and half of the oxygen, and then we have water. So this is my reduction. The electrons goes from here to here. Now if they straight ahead go to oxygen, it's a very high energy difference. So this energy difference is of course something which I would like to convert into ATP at the end. Again, I want to generate energy. However, this energy difference is so high, if I do that directly, I have no chance to put my hands on this high energy difference. So this doesn't work. So the trick now is, what is done in the cell, I split this energy transfer. In other words, the transfer of a substance of a high electronegativity, so this is NADH, onto oxygen, which has the highest possible electropositivity in this case. So I have the highest potential. So I split these in smaller chunks so that I transfer that sequentially to substances with a higher electropositivity. I transfer it in the first step onto coenzyme Q, in the next step on cytochrome C, and this is what I do. So the electrons go sequentially down, and then I can always take out the energy of this step. I don't lose this energy by producing heat. I would lose that if I use this complete step at one step. I just conserve it. And this, of course, doesn't happen freely because electrons cannot freely float around. So this happens in a membrane-coupled process. Yeah, and this is respiration. So at the end, this chart here is what's happening in respiration. So welcome to respiration. And of course, you might have already talked about the respiration chain. So the respiration chain then will look a bit more sophisticated. But at the end, what's happening in the respiration chain is exactly this reaction. This is what's happening in the respiration chain. And of course, in more detail, you have then these complex ones and so on. So you transfer the electrons to complex one, and there are a couple of steps in between. So it's not the coenzyme Q and cytochrome C step. It's not complete. However, for us, we're happy with that. And what you do is, at each time when you transfer the electron to a substance with a higher electropositivity, you use the energy from this potential to pump protons. So at the end, what you do is you pump protons, and the energy what you generate is you generate an proton gradient. And this is the electromotive force what you have. And this is what we learned is also a way to generate ATP. So how, this is the detail you don't know, need to know all this details. This is something you will certainly forget. This is the important thing. Remember this. And that you, of course, build up this protomotive force, and then you use a special enzyme, which is an ATP synthase, which can, from this protomotive force, then form ATP. So this is kind of really rotating and so on. It's a cool enzyme. This is really, the motion is in this enzyme. So if you look at animations, of course, also for biochemists, this is a dream, such a molecule. But for us, it's important. This enzyme can couple ATP synthesis to a protein gradient. By the way, what I've forgotten in the citric acid cycle, also in the citric acid cycle, most of the enzymes are reversible. So if you conserve the energy, this means that the delta G of these reactions should be close to zero. Otherwise, you don't conserve the energy. And therefore, it could go in both directions. There are some exceptions, like the citrate synthase and the alpha-ketoglutarase dehydrogenase, for example, but most of the enzymes there are reversible. And the same is true here. So if you have a proton gradient, and the protons get in, and you couple that with your ATP synthase, you produce ATP, but it could also go in the other direction. So you could also generate a proton gradient using ATP synthase. That's important for example, bacteria. If the motion of flagella in bacteria uses not ATP, it uses the protomotive force. And in general, this looks like a flagella, so you have a rotation then. Therefore, if you're anaerobic, for example, you cannot only produce this gradient if you then burn ATP for doing that. Yeah. Okay. We produce in the citric acid cycle NADH, but we also produce FADH. You remember this is in the oxidation of succinate to fumarate, but there we just form this double bond, and I said the electronegativity of this transfer is not as high enough, so therefore you can just produce FADH. And you see that this FADH is fed in at a different point because it has a lower electronegativity, so therefore it's getting in there, and therefore you have less protons which are being pumped. By the way, this FADH is already membrane coupled, so this process happens at the membrane. So the oxidation of succinate to fumarate is a membrane associated process. Just for the sake of completion, that's not something you need to know by heart, it's just to complete the story. Okay, so you can produce 10 protons, and this is around about 3 ATPs because you need 3 protons to produce 1 ATP, and from FADH 6 protons you can produce around about 2 ATPs. So if we take that together and make the overall energetic balance for our glycolysis and glucose oxidation, you see that our glycolysis, we produce 2 ATPs from substrate level phosphorylation, and we have produced 2 NADH molecules, and these, as we said, correspond to 6 ATPs, so we have in total 8 ATPs from glycolysis. If we couple that again to respiration and the citric acid cycle, we have the 2 times 4 NADHs plus 1 FADH plus the GTP, which is of course equal to ATP, so we have 1 GTP, 12 ATPs which we could form from NADH, and 1 ATP which we can form from FADH, sorry, 2 ATPs, and this also for 2, so we have 30 ATPs coming from the citric acid cycle and 8 ATPs coming from glycolysis. This makes in total 38 ATPs. So you see the difference is enormous. If we use this electronegativity and oxidize NADH and can couple that to transfer onto oxygen, we can produce a lot of energy. So we can produce 38 molecules of ATP per glucose. Remember, if we just have glycolysis, we just produce 2 ATPs. That's an enormous difference. And what is interesting is the efficiency of this glucose oxidation. So you can compare that. So you can of course chemically measure what is the energy of combustion of this reaction. So if I burn glucose, you can measure how much energy is in this glucose molecule, and the delta G is minus 2870 kilojoules per mole. And the energy of, at the end, this is conserved then, transferred onto the ATP, and the energy of hydrolysis of 38 ATP molecules is minus 1208 kilojoules per mole. So this means that the efficiency of this overall process is 42%. And this is better than nuclear power plants, coal power plants, and so on. So this is a very, very good efficiency. And this is the reason why you have to do that in such a complex looking way, because you need to conserve these energies. You need to put these steps always in chunks which are ideal for ATP transfer. So you don't want to generate exothermic reactions where you have then a delta H of minus 500 kilojoules per mole. You want to come close to these minus 31.6 kilojoule per mole you need for the formation of ATP from ADP. And these chunks you always generate, and then you optimally conserve the energy. And that's the intention of this catabolic pathway. This has been optimized for efficiency, and this then dictates more or less how the process looks like. This is evolution. So you don't need God and Jesus drawing around. You just need a lot of time in order that such a pathway can evolve. Now just having a look whether we have questions. Yep. So, why isn't acetyl-CoA present in glycolysis if it's so powerful? We have fermentation, remember, and we have respiration. Respiration means that we need an external electron acceptor. So an external electron acceptor is, for example, oxygen. If we have oxygen, we can oxidize NADH. Everything is cool. However, if we have no oxygen, if we have or another external electron acceptor like nitrate or whatsoever, then we need to get rid of this NADH. So this is then our problem. If we produce acetyl-CoA, then we have the problem that this comes from pyruvate. It's an oxidative decarboxylation. Then we produce another NADH. We don't produce ATP directly in this step. We produce another NADH. So we need to get rid of this NADH again. So, and if we have fermentation, our intention is produce as little NADH as possible and produce as much ATP as possible. So in a fermentation pathway, the citric acid cycle is simply not possible because there you produce tons of reduction equivalent. You will have no chance to get rid of them. So therefore glycolysis, if you have fermentation, stops kind of at the level of pyruvate, and then you have one oxidation step where you produce NADH. This is the oxidation step of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and then you need to get rid of your NADH. So that's the reason why this makes no sense in your citric acid cycle. Reduce cofactors, this is okay. It's a reason why it's membrane coupled? In my opinion, no, but it's simply the process happens at the membrane, so it's kind of smart if you are already at the localization. So in this case, I think the FADH is not really used for many, that's just a speculation by the way, anabolic reaction. So it's no need to have it freely diffusing around in your cytoplasm. So couple it to the membrane where the electron transport happens anyhow. But it's more a philosophical question. I cannot really respond to that. Reversal will take place at a random step in metabolic pathway. What its purpose? So the reversible step per se is not of a purpose, but overall, if you want to get this efficiency, so if you want to conserve the energy as good as possible, this means at the same time you have to put in as many reversible steps as possible, not because you put in a reversible step, but you put in a step which only produces a low delta G overall. So if you, for example, if you have now let's talk about, for example, 1,3-bisphosphoglycerate. If you have a phosphatase forming from 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphoglycerate, so this phosphate which is bond with a phosphoanhydride group just leaves via hydrolysis, this would have a delta G of, I don't remember exactly, but maybe 35, 36, minus 35, 36 kilojoule per mole. This is not reversible. This is an irreversible reaction. However, as soon as you couple that to ADP formation requires to form the phosphoanhydride bond forms 31, minus 31 kilojoule per mole. This means then a coupling of this reaction to the other reaction, then your delta G goes up from minus 36 roundabout kilojoules to minus 5 kilojoules. So it's still exothermic, but just mildly. And this now is a reversible reaction. So that's the reason there why this is reversible, because you keep the delta G close to zero, because then overall, because you couple the energy to another reaction, then your delta G goes close to zero, and this makes the reaction reversible. It's a very good question whether it's a good idea to make all reactions reversible. There we come then more to systems biology and so on, to the complete design of metabolic pathways, because one thing is energy yield. So here we talk about energy yield. So this the glycolysis and citric acid has certainly been optimized kind of for the energy yield. However, at the same time, we can talk about fluxes. So what is the amount what you can put through? So on the first side, it looks, if it's aerobic, you would say always you would go for ATP production via respiration, because you can produce way more ATP in this reaction. However, for example, yeast, if you have a high amount of sugar around, yeast does things like the Crabtree effect. We will look at that in more detail in bioprocess engineering. So it does, despite the fact that it can do respiration, despite it does a fermentation reaction, so it produces ethanol from glucose, you only get out per molecule of glucose 2 ATPs in this case, instead of 38. Why should the yeast do it? Because per time, if there is enough glucose, yeast doesn't care about energy efficiency. Yeast just cares about how many ATP can I produce per hour. And if you tailor everything to make the flux as high as possible, it can be that just with a fermentation and a high throughput, you produce more ATP per time than with glycolysis, where you have this high amount of reversible reactions. So it depends on the condition, and this is interesting to check that in systems biology, what is best. Okay, this was an aerobic respiration. So if I say there is an aerobic respiration, we have also anaerobic respiration processes. For aerobic respiration, by definition, we have oxygen as an electron acceptor, but we can use also other electron acceptors. As examples, we can use, for example, protons. We can use carbon dioxide as electron acceptors, producing acetate or producing methane, as you see later on, sulfur, carbon dioxide, methane, and so on. So at the end, we can use a respiration just means external electron acceptor. Oxygen is cool because you remember in this redox tower, oxygen was at the very bottom. So the energy potential we can produce with any electron donor is highest if we have oxygen on the bottom. Yeah. And the electron, the potential difference at the end, this is our energy. However, if oxygen is not present, we can also take something else. And for example, carbon dioxide and so on. Of course, the electronegativity and the energy which can take out differs then, but still it's better. If I can do respiration, the energy yield will be higher than with just fermentation. So if there is a limited amount of glucose there and it's all about energy efficiency, I have an advantage if I can use another external electron acceptor. Can we do that? No, we can just grow on oxygen. Yeah? So there are two things. Dissimulative metabolism, this is if I produce energy. So I do respiration. So I take up, for example, nitrate in order to use that as an electron acceptor, and I spit out then nitrate. So this is dissimulative. But of course, if I grow, I also need nitrogen. So cells will also take up nitrogen in order to put that into biosynthesis. And this is then called an assimilative metabolism. So assimilative is everything where you tailor your molecules to biosynthesis. And all these molecules here you could use also for biosynthesis. But if we talk about dissimulative metabolism, we just use that for energy. So if you have this pair of ferric and ferrous iron, this goes in and out. It's not used for growth in a way that you put it in your molecules. It's just used as an electron acceptor. And as soon as your electron is put on and you have your Fa2+, it's getting out. So this is the difference here. Okay, now let's look at certain reactions we have. One example is nitrate reduction, and this is the dissimulative reduction of nitrate. And the first step is we go from nitrate to nitrit, and the complete step is from nitrate to nitrogen. And this is a very important process, because so what's happening for the bacteria there? For the bacteria, they produce energy because they use in each step, they use these intermediates as electron acceptors. And at the end, you have the production of nitrogen. But if this happens in your soil, and if you have in your soil nitrate, and the nitrate is of course something your plants want to take up in order to grow. If this is happening in your soil, then nitrate will be denitrified to nitrogen, and nitrogen of course is a gas which leaves the soil. So you denitrify your soil and there is not enough nitrogen inside, and your plants cannot grow anymore. So if you fertilize plants, usually you put in your fertilizer substances inhibiting these denitrifier, that you block this denitrification step in order that the plants can grow better, or you block nitrification. This you can also do the production from nitrate from ammonia, where you use ammonia as an electron donor, and these are then of course chemolithotrophs, by the way. Okay. Now, if you look how this looks like, if, for example, in E. coli, you see this is how it's looking in the kind of, if you use oxygen as an electron acceptor, and you see if you then go to another electron acceptor, just this complex is changed to the nitrate reductase complex, and the remaining complexes stay the same. So you can kind of, you have then your kind of modular system what you use depending on the electron acceptor. Of course, this is nitrate can be often used by bacteria as an electron acceptor. However, this full denitrification, for example, this is done by specialists. Yeah, you see, and then of course you need way more specialized enzymes. And if you usually live in oxygen and you say sometimes I might use nitrate, then you think of, wow, is it really worth it to have all these other enzymes in order that these 5 times a year I need to do denitrification and just to increase my energy yield, that I have all these stuff in my genes. Then you say, no, wait, I just put this nitrate reductase and I'm happy. Then I just can produce nitrate, but it's better than nothing. Yeah. So now, very important processes are acetogenesis, methanogenesis, and again, this is anaerobic respiration. So the major part of methane, which is produced in nature, is produced using this process. So on the left, so we have hydrogen, which is produced by acetogens, and then we have our carbon dioxide getting in this case, of course, it's solubilized as the salt or the acid, and then we produce methane, and this is an electron transfer. So we use carbon dioxide as an electron acceptor, and we can produce quite a bit of energy here. And we can do the same if we, instead of methane, produce acetate. Usually we have approximately the same amount of energy we can produce here, and this is the process. It's very special because this process, as you remember from microbiology, from our basic lecture, this is only happening in archaea. So this is not happening. Bacteria cannot do that. Only archaea can do that. So you start with carbon dioxide, you end up with methane, and of course you have a couple of steps in between, and these steps are sometimes use special enzymes, special cofactors, and so on. So this is kind of a very special electron transport chain, which differs tremendously from the standard one where you use oxygen as electron acceptor. By the way, here, what you pump here, you pump not hydrogen, you pump sodium. So you, in this case, you generate a sodium motive force in the case of methanogenesis. This delta G looks cool, so it looks like, wow, you can produce a lot of energy. However, of course, you remember that this delta G depends on the concentrations. You remember we had this Nernst equation where we had the concentrations in, and here in these reactions in nature, usually the carbon dioxide concentrations are not the problem, but the hydrogen concentrations are very low, and therefore the energy you can get out in this reaction is way lower than it looks like here. Yeah, so this is just for the sake of completion. And this is then, if we look at methanogenesis, what's happening there in the complete process. So methanogenesis means biogas production, either natural biogas production or in my biogas reactor, which is a very important reaction, of course, in the case of a change to renewable energy, because you can sustainably produce your biogas steadily. So you start with your polymeric feedstock, and you see it's not a single step reaction, there are multiple steps here. So the first thing is, if you start with your polymeric feedstock, you have an hydrolysis step here, just looking whether I'm in the way now. And then you have a fermentation, and then you produce your fatty acids, small chain fatty acids, like propionate, butyrate, lactate, and so on. And you either produce then directly from your monomers acetate, here we are, or you produce from these substances acetate and hydrogen. So these syntropic acetogens, they form acetate, but they come from substances which are already quite oxidized. So in order to, in this case, they have to produce also hydrogen to form acetate. And now they are called syntropic because this process only works if your hydrogen concentration is very low. Again, in this case, it's unfavorable if you have a high hydrogen concentration. So this means that they live together with the methanogens, which consume the hydrogens. And this is called a syntrophy because this process only can happen if the syntrophs are in close viginities to the methanogens. And the methanogens then either from acetate from the methane, this is kind of the main process, or from carbon dioxide and hydrogen, and at the end you have methane, and this is the anaerobic digestion chain, and this is what at the end then comes out. We're done with respiration. So see, it's simple. At the end, put the electrons from NADH on your oxygen, take the energy in between, which goes to the proton gradient, make ATP. That's it. That's respiration. Welcome to the club. Thanks again for your attention, and goodbye.